Good day, everybody. This is the first webinar out of a series of 4 webinars to present the ProteOn XPR36. And this webinar is titled An Introduction to the ProteOn XPR Surface Plasmon Resonance Interaction System. And in this webinar, we will present the applications, the main features, and the capabilities of the ProteOn XPR36. So the ProteOn XPR36 is an SPR-based instrument which allows you to measure in real time and label-free any biomolecular interactions. What is actually SPR? SPR stands for Surface Plasmon Resonance, and it's actually an optical physical phenomenon that was discovered in the 70s, initially used to detect gas phases. But 15-20 years ago, it was harnessed for the detection of biomolecular interactions in the liquid phase. And actually, this optical phenomena is used to measure the refractive index changes over a gold layer, a gold surface, which is on top of a chip. And these changes in refractive index are proportional, directly proportional to mass concentration changes over this chip. And this allows you to measure the interactions of biomolecules without the labeling and in real time, and therefore it allows the direct measurements of kinetic binding constants. So what are the questions that you can answer using SPR? First is, how specific is the interaction? Or in other words, you have two binding molecules which you want to know if they bind, yes or no. And if they bound to each other, you can determine the association rate constants, or in other words, how fast is the interaction. Once the complex is formed, then you can answer and determine the dissociation rate constants, and therefore to know how stable is the complex. And from these two parameters, or to make a direct measurement, you can determine the affinity constant, the big KD, the affinity constant of the interaction. And the types of data analysis that you can do with the ProteOn XPR is, as we said, first, yes, no binding. Do these two molecules bind each other? Then, to determine the kinetic binding constants, the Ka and the Kd, the association and dissociation rate constants. As mentioned before, you can determine the affinity constant, the equilibrium constant. Also, one can determine the concentration, but the active concentration of a protein in a solution. And this is unlike just measuring total protein concentration in a vial using, let's say, 280 nanometer, which will tell you how much total protein is in your sample. But using the ProteOn XPR, you can determine the active concentration of your protein. And this is mainly important for biopharmaceutical companies which manufacture therapeutic proteins, and they need to know what is the active concentration of their compound. And because it's a functional assay, it allows you to determine only the active concentration of it. You can also do thermodynamic analysis by measuring the interaction at different temperatures, and from there to extract the free energy, and the enthalpy and the entropy. And these parameters actually explain or shed light on the magnitudes of the binding kinetic constants. So basically, you can measure the interaction of any biomolecule of interest, so protein peptides, antibodies binding to antigens, protein with small molecules, DNA binding to proteins, DNA-DNA interactions, and whatever is considered a biomolecule, you are welcome to test and try if it binds to its partner. And the different applications that one can do are various, and this is just a partial list. One can do screening of antibodies. If you have supernatants of different monoclonal antibodies and you want to rank based on their affinity, you can characterize the antibodies to determine the kinetic binding constants, to do isotyping, to do epitope mappings, of course, to screen libraries of small molecules interacting with the target protein, as mentioned, to do concentration analysis, and fundamental or basics proteomics work to determine proteins complex formations or dissect the interface of two proteins binding to each other, and so on goes the list. So how does it actually work? How does the binding event monitored on the ProteOn XPR36? So in any typical assay, you have two binding molecules. One would be covalently bound or captured on the surface, and we call this molecule the ligand. The other one is flown over or injected over the ligand, and we call it Analyte. And you should remember that there is always a continuous flow of buffer or liquid over the chip where the ligand is immobilized. So always either running buffer is flowing over or the Analyte is being injected. So this is a cartoon. This is a cartoon showing how the data, the output of the data in real time. So in this upper panel, it's a cartoon of the ligand immobilized on the chip, and here in the lower panel, this would be the output as would be seen on the user's PC. So at the first stage after the immobilization, just running buffer is flowing, so the signal is zero. There is no change in refractive index. It's just the running buffer flowing. Then when you're ready with your sample to inject the Analyte over the ligand, the Analyte starts to flow and starts to bind to the ligand. And because mass is accumulating near the chip surface because it is binding to the ligand, there is a change in refractive index, which will give a response that will be shown in real time. Once the Analyte injection is over, running buffer is back, and you will see the dissociation of the Analyte from the ligand. And this output is what we call the sensorgram. And from this sensorgram, you can determine the binding or the association rate constant, the equilibrium, and the dissociation rate constant. And one should always remember that this sensorgram is an output of one Analyte concentration only. And to determine reliable kinetic constants, one should always inject several Analyte concentration for having several sensorgrams. So just to summarize again, this is the sensorgrams. In the x-axis, you will see the data in seconds because it is a real-time measurement and not an endpoint measurement. And in the y-axis is the response unit. So this is one sensorgram coming from one Analyte concentration. And in this sensorgram, you can extract the association rate constant, the dissociation rate constants, and from these two, or directly, the equilibrium or the affinity constant. So as I mentioned and emphasized, one Analyte concentration will yield one sensorgram, but to determine reliable kinetic constants, you need several sensorgrams, 4, 5, or 6. And currently, most of the instruments available in the market are operating in a sequential mode, meaning that one Analyte concentration is injected, then you need to regenerate the surface to remove all the Analyte that is bound to the ligand, and then inject Analyte concentration 2, regenerate again, Analyte concentration 3, and so on. And this is the advantage of the ProteOn XPR, which allows you to do all those things in parallel. And this is what we call the one-shot kinetics. So the ProteOn XPR36 has 6 microfluidic channels which function in parallel and independently. So in the first stage, you can immobilize up to 6 different ligands in parallel, or the same ligand at different conditions, or you can use today channel 1 and 2, tomorrow channel 3, 4, 5, and next week channel 6. So you have the full flexibility of how much to immobilize and what to immobilize. When you're ready with the immobilization, you proceed to the next step to inject the Analyte, the different Analyte concentrations, and now you prepare 6 Analyte concentrations, and in one injection you'll get 6 panels, each one consists of 6 different concentrations. So this panel here is the outcome of those 6 analyte concentrations interacting with ligand number 1. And this panel having 6 sensorgrams are the same 6 analyte concentrations interacting with ligand number 2, and so on. So basically in one injection, or what we call one-shot kinetics, you can derive and extract up to 6 sets of Ka, Kds, and affinity constants. In this example, in channel number 6 is empty because it was used as a reference. Each experiment has to have a reference spot, but using the ProteOn XPR, you can actually mobilize on all 6 channel ligand and still having a reference, which I will be showing in the next slide. So this is an upper view from above of the chip surface. So you can immobilize 6 different ligands in this direction, or what we call in the ligand orientation. And then when you inject the analyte, 6 analyte concentration in the analyte direction, these analyte will flow over the entire channel. But if you look here in this area, there is no ligand because the ligand was immobilized in these channels. But in this spot, there is only the chemistry or the chemical layer of the chip. So this spot will be served perfectly as a reference. So basically, each 36 spots, each one out of the 36 spots will have its own interspot, what we call those referencing spots or interspots. And these reference spots or interspots are excellent referencing because they are very close to the measuring spot where the interaction takes place. So the proximity provides very good referencing. So just to summarize the capabilities, so we say that the ProteOn XPR36 has 6 independent channels, once in the ligand orientation flow in this direction, and when you inject in the analyte orientation, it will go in this direction, in the horizontal direction, and thus creating a matrix of 36 active spots, interaction spots, which are measured simultaneously by imaging. And remember the interspots that are used as referencing spots for each one out of the 36 spots. So, and it's not only just an array of 36 spots, it's the flexibility that is allowed by the ProteOn and by the special and unique microfluidic system, because for the ligand, for example, for the ligand stage, you can immobilize 6 different ligands, or you can immobilize the same ligand but at different densities to determine which density or which channel has the best amount of ligand, or you can test the same ligand but with different conditions, different buffers, different salt conditions. And when you are ready for your analyte stage, you can inject the same analyte at different concentration, or what we call the one-shot kinetics, or you can do screening assay to inject 6 different compounds at the same concentration and to screen which compound out of those 6 binds to your ligand, or you can also do only 2 compounds, each one at 3 concentration, to determine their kinetic binding constant. So you have the full flexibility to design your experiment the best way is convenient for you. So just a quick tour over the instrument. This is a front view of it. Here in the middle of it, there is the Autosampler where you put your samples. On the right side is the buffer compartment. Remember, there is always a continuous flow of running buffer, and you have place for 2 running buffer, which you can switch between them easily, either by those buttons here below or from the software. And this is very convenient because if you need running buffer A for the immobilization of ligand and running buffer B for your analyte, you can easily switch between them or enter a command in the protocol when you set it up. On the left side, there is the chip, where the chip comes in with all the optics behind it, and there are of course status LEDs of the instrument which will indicate about the progress of your experiment and the instrument status. A deeper view into the autosampler. The autosampler can accommodate 296 well plates, either regular one or deep one, each well containing 2 ml, or you can also use a rack with those vials which have a cap, and they will be pierced by the needles to take the samples. So you can choose wherever is convenient for you. Here below you can see again the 2 bottles of the running buffer and the syringes that drives the 6 channels simultaneously. So one set of 6 syringes is for the sample and the other one is for the running buffer. As for the chips, the chips comes inside a cartridge, and the cartridge has a barcode on it which stores all the information of your experiment, all your experiment results and data and logs are stored on the software, which the barcode reader will identify it. So the cartridge is inserted into the instrument, and the instrument will take out the chip from the cartridge and put it in the right place inside the optical path. So what chips are currently available? Currently we provide 4 different functional chips, including 2 other chips that are used for maintenance and for cleaning the instrument. So the first chip is what we call GLC, or General Layer Compact. It contains carboxylic groups to immobilize your ligand using the amine coupling, which we will describe a little bit later. And this chip is mostly suitable for protein-protein interaction, or in other words, more specifically, when the analyte is big enough, so this chip would be suitable. The second chip is GLM, which has a medium capacity. It also contains carboxylic groups, but more of them, more than compared to the GLC. And this chip is mostly used when your analytes are smaller, like peptides or small molecules, so you can bind or immobilize more ligand on your chip, and therefore to introduce more binding sites, and therefore more small molecules would bind to the surface, generating a bigger signal. The third chip is a GLH with high capacity, and with this chip you can even immobilize up to 20,000 R units of ligand, and this is optimal for very small molecules. And you can see in this example, it's the interaction of an antibody with a molecule of a size of 241 Dalton. So the ratio is really challenging, but the sensorgrams are very significant and nice. The fourth chip is an NLC chip, which is actually a GLC chip covered with neutral within layer, and this is mainly used when your ligand is biotinylated, like DNA molecules or other proteins that you can biotinylate. And when you inject your biotinylated ligand over this chip, it will be captured by the avidin, forming a very stable ligand layer because of the nature of the avidin biotin binding constants or affinity. So how does a basic SPR experiment looks like? So it has 2 sections, of course. First, you need to immobilize your ligand, and then once the ligand is immobilized, you inject your analyte to measure the interaction. So for the immobilization, you need first to activate the carboxylic groups of the surface layer, then to flow over your ligand, which will interact a covalent amide bond with the activated carboxyl groups. Then you need to deactivate all those activated carboxyl groups that did not interact with the ligand. And here you are done with the immobilization. In the next step, you will inject the analyte to monitor and measure the interaction. And finally, once you collect all the sensorgrams, you need to process the data and analyze it in order to determine the binding constants itself, the association and the dissociation rate constants. So a deeper view into the immobilization stage. The immobilization of a ligand can be done in two different ways, two different principles. The first one is what we call coupling, which is actually a covalent or a non-reversible binding of your ligand to the surface, and this is done by using functional groups of the ligand, like amine groups presenting in the N-terminal or mainly on lysines. So primary amine groups on the ligand will interact with the activated carboxyl groups, and the coupling procedure is a random coupling because any primary amine groups on the ligand will interact with the activated carboxyl groups. Coupling is very efficient. You can immobilize a lot of ligand using this way. And you can also, another nice and elegant way is to perform or create a cysteine or a cysteine binding. If you have a peptide, you can engineer a cysteine as the first amino acid, and on the other side to introduce a cysteine on the chemical layer, and then it will form a disulfide bond, which will be very stable. The other method is capturing, and this is a non-reversible binding of your ligand to the surface, and it's actually taking the advantage of a specific recognition site, like, for example, a monoclonal antibody that will capture a certain molecule out of your sample, and this is mainly used when you have supernatants or lysates or serum, and in this serum or supernatants there is a mixture of many proteins, but you want to capture only a specific protein out of it. So if you immobilize a monoclonal antibody on the chip, it will capture only those molecules that you desire. And the advantage of capturing is that it will create a homogeneous surface. It's not random. The surface will be, all the ligands, the surface will have ligands that would be presented to the analyte in a homogeneous way. But one thing to take into account, that because it's a reversible binding event, your ligand might decay from the surface as it is not a covalent binding event. So this is how an immobilization profile looks like. The immobilization is actually the coupling of a ligand to the surface actually has three stages. First, you need to activate the carboxylic groups. Then, after the carboxylic groups are activated, you inject your ligand on the surface, and then you need to deactivate all those remaining carboxylic groups that did not interact with the ligand. And the immobilization level is actually the signal after the deactivation compared to the signal to the baseline before the activation. And this will tell you how much ligand was immobilized. So this is for one ligand on one channel, and this is how, in this slide you can see how it will look like on the instrument when you immobilize 6 different ligands on the 6 different channels. So here you can see the immobilization of ligand 1 on channel 1, and here the immobilization of ligand 2 on channel 2, and so on. So you do it, the immobilization of those 6 different ligands in parallel. Just going briefly over the 3 steps of the activation. So, as mentioned, the chemical layer has functional groups that are carboxylic groups, and they will interact with the primary amine groups that are on the ligand. But they will not, of course, interact spontaneously. You need to activate them. So activation is done using a mixture of EDC and sulfo-NHS. It's a well-known chemistry of amine coupling. So a mixture of EDC and sulfo-NHS is first injected over the chemical layer to activate the carboxyl groups. And the advantage of using sulfo-NHS is that it maintains the negative charge of the layer to attract the ligand. In the second stage, after you activated the surface, the ligand should be injected, and to get efficient immobilization, you need to bring the ligand close to the surface. And this is done by diluting your ligand in in a acidic buffer solution having a pH lower than the pi of the protein, and therefore the protein or the ligand will be positively charged and therefore will be attracted electrostatically to the chemical layer that contains sulfur groups coming from the sulfur NHS. So in this way, because the chemical layer is still negatively charged after the activation, you can actually immobilize very acidic proteins like pepsin having a pi of around 2. The last stage after you injected your ligand, you need to deactivate the surface, so any activated carboxyl groups that did not interact with the ligand need to be put off, so the analyte afterwards will not interact with these groups. So the deactivation is done with 1 molar ethanolamine to deactivate all those carboxylic groups. So after you immobilize your ligand, the next step would be to inject your analyte to actually see the interactions. And just few points that are important for the analyte stage. So the analyte concentration that you are preparing, those 6 concentration of analyte are very important. If you inject concentrations that are too high, they will saturate all the ligand binding sites, as can be seen in this panel. If your concentrations are too low, the sensorgram will look very linear and will not reach, none of them will reach saturation. So in an optimal assay, after you characterize all the parameters, you need to prepare 6 analyte concentrations that will bracket the affinity constant. Usually, as a rule of thumb, from 10 times the affinity constant down to 0.1 of the affinity constant, and this will yield good sensograms with good quality. And just a continuation of this, those analyte concentrations that should be around the affinity constant should also be injected for sufficient time. Remember that the binding event are actually a first order interaction or a pseudo first order interaction, meaning that they have an exponential shape. So you must have a curvature into the sensograms in order for the algorithm, for the fitting algorithm, to extract reliable kinetic constants. In this example here, in the wrong example, either the concentrations are too low or the analyte injection is too short. So the sensogram look linear and they do not have any curvature in it, and there are several Kas and Kds that can describe such kind of an interaction. So you will not get reliable kinetic constant after data analysis. So just to conclude, you need to characterize and identify the best analyte concentrations that are around the affinity constant, and to perform long enough injections that will generate a curvature into these sensorgrams in order to get reliable kinetic constants. So when you inject your analyte, you are seeing the data in real time or data collection, and this is how it looks like in real time, in real life, on the user's PC, on the user's screen. So you can see here ligand number 1 in this window, ligand number 1 interacting with the 6 analyte concentrations, and this is ligand number 2 interacting with the same 6 analyte concentrations as we defined previously, the one-shot kinetic approach. And in this channel, the 4th channel, you can see it's just the bulk effect. It's an empty channel used as referencing. And the nice thing in the software is that it allows you to perform real-time referencing, online referencing. So you can define with the software that, for example, channel number 4 or the interspot that we described previously are the referencing, and in real time, as the data is being collected, you can see the reference data, which is the final one, of course. And along the experiment, after you collected many sensorgrams and you perform different injection one after another, you have the flexibility with the software to view and filter out any injection that you like. So you can define and tell the software, show me only analyte injection, show me only ligand injections, and how to group them either by ligand orientation or by analyte orientation, and also you can take out any specific sensorgram out of the viewing. So in this side of the software you have all those filtering options that provide you flexible and powerful data viewing that would be most convenient for your assays. So this is just zooming in of those filters. And after you collected all the raw data and you view it and you selected which data you want to go and proceed for the analysis, you need to process the data. Processing the data means to subtract the reference or to do a DMSO correction if needed, excluded volume correction, and of course to do baseline alignment. The baseline just prior to injection need to be aligned, and also to align the start of injection. So all these commands can be done either manually or automatically using a single button click on all those sensorgrams. So you can actually, using a single button press, process all your interaction. This could be tens or hundreds of interaction. So all those windows shown here will be processed instantly after one click of a single button. And of course you have the flexibility to do this manually for each sensorgram or for each panel or whatever you think is best. And after you process the data, finally you reach the stage of data analysis, which is actually the essence of the experiment, to determine the binding kinetic constants. So this is how, for example, a classical one-shot kinetics data view will look like. You will have 5 ligands, each one of them interacted with 6 analyte concentration. One channel was empty for referencing. And now you need to define, to do data fitting to determine the Ka, the Kd, the dissociation rate constant, and the affinity constant. And what the software allows you, it gives you flexibility to determine how those parameters will be defined. For example, in this example, those sensorgrams that you can see below, it's the same ligand immobilized on 4 different channels, interacting with the same 6 analyte concentration, meaning that all those 4 panels have the same Ka and same Kd. So you can tell the software, determine a Ka and a Kd, only one number for all those 24 sensorgrams, or in other words, global fitting. So the algorithm will only play with one number for Ka and Kd to find the best values for all those sensorgrams. On the other side, on the other extreme, you can tell the software, find me a different Ka for each one of these sensorgrams. So eventually the output will be 24 Kas and Kds. And a nice thing that you have with the ProteOn is something in the middle is what we call grouped. So these 6 analyte concentration that interacted with ligand 1, you can tell the software find the same Ka and Kd for this panel, for ligand number 1, and for ligand number 2 find a different set of Ka and Kd, and for ligand number 3 find a third set of Ka and Kd. And this will be used, of course, when you have different ligands, like different mutants. If you have 5 or 6 different mutants immobilized on the chip that interact with one analyte injection with 6 analyte concentrations. So each ligand surface will have its own Ka and Kd. So you can tell the software, find me a grouped Ka and Kd. And in this example, as I told you, it's the same. It's the same ligand interacting with the same analyte. So once we performed, you can see the results here. Once a global analysis was performed, and once a grouped analysis was performed, and when the results are good, you get the same values, the same Ka when it's global, and very similar Kas when they are fitted grouped, and the same goes for the Kd. So after you obtained the software, outputted the kinetic binding constant, you need to evaluate the results. Remember that the data fitting procedure is just a mathematical procedure. It's a fitting algorithm. It has no common sense, and you need to judge the result and to evaluate if these results are reliable and meaningful. So there are different stages to do that. The first one and the most obvious one is to do visual inspection. Of course, you can judge with your eyes best, and you can see that on the left side, the fit goes very nice through the data, and on the right panel, this sensorgram, the fitting is not so nice because the fit doesn't go very nice through the data. So obviously everyone can tell that this one on the left is a good data fitting. I believe these results, and here on the right, those results are not so good. The second thing that you need to evaluate and to inspect is the magnitude of the constants. And here there is an example, and you can see the output of the fitting, and you can see a Kas of 80 and 70, and Rmax, which Rmax is the signal that will be obtained once all the ligand binding sites are saturated. So clearly these values are not logical. Kas of 80 or 70 do not exist in nature, or not to my knowledge. And this is and once you see such results, you can immediately say that something is wrong. And this is of course because of you can see here the sensorgram. As we saw before, the analyte concentrations were not they were too low, or the injection was too short, so the binding event is still linear, and the dissociation also is still linear. There is no curvature, and therefore the algorithm does not know where the sensorgram will reach saturation, so it finds an Rmax of 1,000,000. So clearly when you see those results, even though the fitting is very nice, it looks nice, when you see those results, you can immediately tell that something is wrong because the values are not logic, and you need to do something to change it. The third thing that you can use to evaluate the results is the plot of residuals. This is a graph that shows the distance between each data point from its corresponding point on the fit. So good results and reliable results, the residual should be distributed normally and with a normal distribution and evenly around the zero line, or actually in other words, the data points should be distributed around the fitting curve, which means just arbitrary or random noise. In this example here below, you can see that the residuals are deviating from the line fit, from the fit of the line of the fit. So something is wrong here. There is a deviation, or you can say there is some systematic error in the sensorgram. Something is wrong here. And the last thing, two things that you can determine, evaluate in order to tell if your results are reliable. The first one is the chi-square, which is actually the sum of those residual, the average sum of those residual, and of course one should expect a low chi-square, and the lowest chi-square that can be available is around a value that is close to the noise of the system. And the last parameter is the standard error. The standard error is not the standard deviation. Standard deviation is to perform the experiment several times to extract different Kas and then to do average and standard deviation. But the standard error is actually a parameter that is extracted from the fitting procedure, and the value of the standard error tells you how reliable the fit is. So here are shown two examples. So in this upper graph, you can see a parameter on the x-axis. It's the parameter value, and you can see that if the parameter value or magnitude is a little bit changed, the sum of square are increasing tremendously. On the other hand, if a parameter, in this lower example, the parameter is poorly defined. Why? Because you can change the parameter magnitude. It can be, let's say the Ka can be 1,000, 10,000, 100,000, and still the sum of square does not change significantly, which means that you can never know if what is the real magnitude of it. Is it 1,000, 10,000, or 100,000? So good data fitting will have a low standard error, which means that the parameter is well defined, is robustly defined. And usually, as a rule of thumb, the standard error should have a magnitude of 10% of the parameter itself. So this was the first part to describe the ProteOn capabilities and just tips and hints on how to perform a basic SPR experiment with all those stages of it. And in the second part, I will describe several applications that were done and collected with the ProteOn XPR36. So the first one is an example of an assay development using the parallel processing of the ProteOn XPR, the 6 channels that runs in parallel. So we determined the binding constants of Interleukin-1 receptor with Interleukin-1 and its antagonists. So we got the Interleukin-1 receptor for the first time. We didn't know which conditions are best to immobilize it, so we used the 6 channels. In each channel, we used a different pH of the acetate buffer, and in one time we mobilized on 5 channels. The 6th channel was left empty for referencing. So you can see here the immobilization levels zoomed in for the different pHs. And then we injected the Analyte, just the Interleukin-1, to see the activity. Because remember, it's very easy to, not very, but usually it's easy to immobilize the ligand, but the tricky part is to maintain its activity. So you can see in this graph a summary of this assay. So the ligand density is the left y-axis. You can see that at pH 5, the highest amount of ligand was immobilized. But surprisingly, or not surprisingly, the ligand using pH 5.5 was most active, which means that although less ligand molecules were immobilized on the chip, still more of them were active than compared to pH 5. And this is a nice way to use the 6 channels to characterize in one time. You have a new pair of proteins. You want to determine their kinetic binding constants, and you have no clue of, just the first time, you have no clue of what would be the best condition. So you can test 6 different conditions in one time on a single chip in just a few injections, and you can get the answer what are the best conditions, and of course, together with that, to have the results. So here you can see on the left, the Interleukin-1 interacting with the 5 Interleukin-1 receptor channels, and we did global analysis for Ka and Kd. And here on the right, it's the antagonist, and of course the antagonist has a lower affinity than the Interleukin-1 itself, which would be expected in a biological system. The second example is the development and the discovery and development of therapeutic antibodies using the ProteOn XPR36. And this is a long project that we did together with a small pharma company that they are manufacturing or discovering and developing anti-cancer therapeutic antibodies. And the starting point is a phage display library of phages displaying a Fab fragment. And it's a big project that is composed of several or 4 sub-projects, which are described in this slide. So the first project is just to screen for finding the best Fab fragment having the highest affinity for the antigen. Then this Fab fragment with the best affinity is assembled to or fused to an Fc part of an antibody to get an entire immunoglobulin. After that, there is a step or third project of affinity maturation to increase the affinity of the selected antibody. And the final step is to introduce mutation in the Fc region of the antibody for modifying a complementary process or the ADCC or CDC. So just before we started, we did a kind of validation. So we selected a commercial therapeutic antibody, Herceptin, manufactured by Genentech. And this is a shelf, a drug available on shelf. So we purchased the Herceptin antibody and we immobilized we captured the antibody and then injected different concentration of HER2, which is the antigen, the target antigen. We determined the binding constant, the Ka and Kd, to validate the system. So I will now describe the work done in those 4 sub-projects. So for project number 1, if you remember, was just to screen out of the library the best Fabs having the highest affinity. So in the first step, we just immobilized a Fab anti-Fab on the chip, covalent immobilization. In the next step, we captured 5 different Fab soups. Those are the Fabs to be screened, Fab fragments to be screened. So in one injection, you can capture 5 different soups. In the 6th channel, we injected buffer to be used as a reference. In the 3rd step, the microfluidic channel is changed to the Analyte direction, and 5 concentration of antigen are injected over the captured Fabs to determine the kinetic binding constant. So this is a classical one-shot kinetics analysis. And after the kinetic binding, those sensograms are collected, there is a regeneration step to remove the antigen and the Fabs from the capturing Fab in order to capture now Fab number 6 to 10 and so on. So here you can see just representative sensograms of the different Fab fragments. You can see how the affinities are varied. And most of the Fabs that were screened had affinities of 1 micromolar, but we could identify and did repetition. We could identify 2 populations having affinity of 5 nanomolar and 80 nanomolar. So these Fabs were, of course, picked and selected for further analysis. And before that, before we proceeded with these 2 Fabs, we did a validation to eliminate any possibilities of artifacts. So we captured those Fabs in 3 different ways. In the first step, in the first way, the Fab anti-Fab, was covalently immobilized on the chip, and as the usual experiment, the soup containing the Fab was injected over it, so the Fab was captured by the capturing Fab. In the second channel, the purified Fab was captured by the capturing Fab, and in the third way, the antigen itself was immobilized on the chip, and the Fab was injected as an Analyte. And you can see here the results that they are very similar in all 3 ways, and this is to validate the results that there are no artifacts or no misleading results. So then the second project was to do affinity maturation. So those Fab were subjected to mutagenesis in different ways in order to introduce this mutation for having increased affinity. So you can see those mutants were then screened to determine the affinity constant, the kinetic binding constant, and you can see here for Fab1 how the affinity is now varied, and one can see that mostly is the dissociation rate constant which is affected, and less the association rate constant. And the same was for Fab2. You can see here also the variety of affinities coming from these mutations. So we could screen a big number of mutants, and for example, 180 SUPs could be determined, the kinetic binding constants of 120 SUPs could be determined in 12 hours, which is very powerful. So each cycle is about half an hour, depending on how much association and dissociation time you collect. So, and the third sub-project was, as mentioned, to assemble the Fab fragment to its Fc region to create a whole immunoglobulin. And in this sub-project, there were two goals. One is to ensure that the entire immunoglobulin maintains its affinity like the Fab fragment had, so that the affinity did not change. And the second goal is to measure the amount of immunoglobulin expressed by the cell. So, because the entire immunoglobulin can have an excellent affinity, but if it expresses very low amounts of the antibody, this clone cannot be used for further expression and for purification of the antibody. So this assay was done by covalently immobilizing protein A on the chip on 2 channels. Then in the next step, in step number 2, 4 different immunoglobulins from different clones were injected and captured by protein A. In the 5th channel, a standard concentration of an immunoglobulin of just an IgG was injected to serve as a calibration point. And running buffer was injected in the 6th channel for referencing. And in the 3rd step, the specific antigen was injected over channel 1 in the ligand orientation, and the non-specific antigen was injected over channel 2 to see that there are no cross-reactivity or other things that could happen. So these are the results. You can see the upper graphs are for Fab1 and the lower one are for Fab2. So this is what you see here, the upper left is Fab1, different IgGs, different immunoglobulin. The red curves are different concentration of standard, so one can easily quantify the amount of immunoglobulin expressed by the cells. In the middle panel, you can see the interaction, the specific antigen, and here on the right, you can see the non-specific antigen that does not interact with those immunoglobulin, meaning that nothing has changed during the fusion of the Fab to the Fc portion. And the same goes for Fab2. The 4th project was the modification of the Fc part of the immunoglobulin to introduce point mutation in it, and this is something known in the literature, that there are very specific amino acids that you can change, which will modify, increase or decrease the ADCC or CDC processes. So we performed we had a mutant with 2-point mutation and a mutant with 3-point mutation, and this is the wild type. So we mobilized on the chip the Fc gamma receptor, which is the receptor presented on immune cells, on T cells, and then we injected the IgG and the mutants as Analyte. So you can see here the wild type, and then the interaction of the 2-point mutation antibody and the 3-point mutation antibody, and one can see that both the 2 and 3' mutation antibodies increased the affinity 30-fold, but there was no big difference between the 2 and 3' mutation. And the last thing that we did is another kind of validation. We compared the different construct of Fab1 as a single chain, or as a Fab, or as a complete IgG, and we determined the kinetic constant, the binding kinetic constant of this structure to the antigen to see that there is no change in the affinities and the kinetic binding constant. And you can see here in the table that indeed the results are very similar for each one of the constructs. So the last example is from the world of small molecules. So here we screened a small set, relatively small set of library of inhibitors, small molecule inhibitors, towards 2 different kinases. And the first thing that we did was a kind of a collaboration or with small work done with Merck Pharmaceuticals, and they had a target protein, but they knew that just normal immobilization of it over the chip eliminates its activity because the binding site is lysine rich, and therefore those lysine will interact with the carboxylic groups, and the active site, the binding site of the protein, will be not available to bind these small molecules. So what we did very nicely, using those 6 channels, we tested 3 different pHs for the immobilization, and each pH we used once just the acetate buffer, and once the protein was pre-incubated with an inhibitor, a known standard inhibitor that was bound to the binding site and protected it from being immobilized on the surface. So the protein was diluted in acetate 4.5, for example, together with high concentration of the inhibitor. The inhibitor bounds to the pocket, to the binding site of the enzyme, and therefore it was protected once this enzyme was injected for immobilization. And you can see here very nicely that only those 3 surfaces that were pre-incubated with the inhibitor in the first place, only they maintained activity. Those 3 channels that where the ligand was immobilized without pre-incubation, the ligand lost all its activity. And here again we did global analysis and grouped analysis, because those 3 active surfaces are the same protein, so they should have the same K and same KD. And you can see, because the results are robust and reliable, you get similar values even if you do global analysis compared to grouped analysis. So then we continued to our experiment, and we had 2 kinases. One was P38 wild type, and the other one was an ERK2 kinase. And we adopted the same tactics, the same procedure here. So both enzymes were once immobilized directly and once immobilized with pre-incubation of a well-known inhibitor or Analyte. So on channel 1, the P38 was just immobilized. On channel 2, the ERK2 was immobilized. And on channel 3, the P38 was immobilized with pre-incubation. And here as well, ERK2 was immobilized after it was pre-incubated with ATP. And you can see the results for the P38. Without pre-incubation, all the activity was lost when we tested 2 different analytes. But when it was pre-incubated with SB203580, it was active to bind the same analyte that it was pre-incubated with it, but also with a different analyte having a molecular weight of 360. And you can see the kinetic binding constant in this table below. For the ERK2, there was no actually need for the pre-incubation because it was active even when it was even if it was not pre-incubated with ATP, and the kinetic constant are very similar, even with pre-incubation and without pre-incubation, meaning that the binding site is not blocked upon the immobilization procedure. So once these two kinases were immobilized, then we characterized the immobilization conditions. We then started a screening assay, and a screening assay is performed by injecting a single concentration of each compound. In each injection, 6 different compounds, and the signal level is being recorded using a report point table. The magnitude of the signal for each compound is recorded, and then of course it is normalized according to the molecular weight, because as you remember, the response unit is proportional to the mass of each compound. So obviously a bigger compound will generate a higher signal, but because it is bigger. So if you divide each of these signals for each compound according to its molecular weight, then you normalize those signals, and then when you get for all those compounds the values for each sensorgram, you can draw a threshold wherever you think is relevant for you. Of course, one can use also a positive control, which is a very high affinity binder, and of course a negative control, an analyte that does not bind, as mentioned, as a negative control. And those that are above those threshold that was defined would be considered as hits, and they will be further subjected to further detailed kinetic analysis. And you can see here those hits that we selected for P38. We did the one-shot kinetic analysis, and these are the names of the compounds with their binding constants. And the same was done for ERK2. All those that were considered as hits were subjected to detailed kinetic analysis using the one-shot kinetic approach to determine the binding constants. And this is just a representative a slide with representative results of few of the 200 small molecule compounds that we screened to give you the flavor of it. So that was it. I hope you all enjoyed it and you understood the capabilities and how the ProteOn XPR36 is working. And if any of you have any additional questions or information that you need, you can contact Dr. Laura Moriarty, which is the product manager of the ProteOn XPR, or you can contact me using these email addresses. So thank you everybody, and hope to see you in the next webinar, which will be the second one out of the 4th, given by Dr. Yasmina Abdish from Pfizer. Thank you very much, and goodbye.